background. Now, I've been on both sides of the table. I've done a PhD and I've done a Viva, but I've also served as an examiner. And a very easy way for a lazy examiner to get to you in your Viva is to sort of look at one of the tools that you've used, whether it's a bioinformatics tool or something in the lab, and say, oh, how does that work then? You know, and they can give you a hard time. There's no script as to how these Vivas work. They can ask you anything they like. So they could come along, oh, you did a homology search. How does homology searching work? You know, and it's one of those things where if you don't have an answer, you know, they just give you a hard time. I mean, you won't fail, but you'll just have a hard time. So that's the reason why I'm providing you with this conceptual background. The other thing is you have to indulge me, because I've been in this game a long time and I like to tell stories from my own experience over the years. So we'll have a meander, but we will actually be leading to the place we want to be, where you would get an understanding of what we're doing as we move through. So let's start with the term homology. So the term homology was first used in biology by the anatomist Richard Owen in 1843. And he was looking at vertebrate fins and limbs. And what he noticed was that if you looked at the vertebrate limb, you could see there was some kind of patterns of similarity there, even though those limbs were doing different jobs. So in some cases they were legs or arms, in some cases they were wings, in some cases they were flippers, but you could see that there in the bones there was, like, coming from the inside out, there was one bone, two bones, many bones, and then 5 digits, if you like. And so there was this kind of underlying similarity there. And Owen developed his own criteria, position, development, composition, looking at how he would define homology. So the idea was that homologous organs are not necessarily similar, and that, or at least that similarity may not be obvious. So it requires a little bit of an expert view to look at what's important and what's not important in determining whether something's homologous or not. And so we can have similar things that are actually doing similar functions, but they're not homologous. So one of the textbook examples is the bird wing allows a bird to fly, an insect wing allows an insect to fly, but they're not homologous in Owen's sense. And there's Owen, you know, slightly scary guy with a very large forehead. You know, it does look like something out of a Victorian Gothic horror movie, doesn't he? Of course, basically everything fell into place. Owen's view was, oh, this was kind of like the creator's plan. That's how you can see the mind of the creator working out in nature, whatever. But it fell to this guy here, Charles Darwin, to come up with this theory of evolution by natural selection and provide explanation for why homology is there. And just to give you you know, basically lots of ideas. He wrote The Origin of Species. There's his collected works on my bookshelf at home. And I did write a book about this just over a decade ago called Rough Guide to Evolution, which is no longer in print, but you can still buy it on Amazon, although the price seems to have gone up. At one stage it was very cheap, and now it's gone up again. And, you know, just to give you an idea of what Darwin was about, just to remind you, because, you know, nothing in biology makes any sense except in the light of Darwin's evolution. Darwin wrote his first ideas about evolution in a series of notebooks that he kept on the Beagle and elsewhere. And his very first sketch of an evolutionary tree is shown here. This isn't the sketch that is normally reproduced, but this is actually the first one he produced here, where he showed that this kind of dotted line were extinct forms, and then these branches of a tree there showing extant forms. And if you're interested in any more about this, I've actually given talks that go into a lot of detail about the Darwin's ideas and how they evolved in the notebooks. And in fact, just as an aside, you all think of Darwin as this kind of old man, grandfather figure. But in the notebooks, you see Darwin as a young man full of hormones, and he says all sorts of rude things about himself and about women and sexuality and all sorts. So it's quite interesting to look at those notebooks. Here's just one quote that comes out of one of the notebooks, which I think actually speaks very much to the modern kind of zeitgeist. If we let conjecture run wild, then animals are fellow brethren. Pain, disease, death, and suffering, and famine are slaves in the most laborious works, are companions and amusements. They may partake from our origin in one common ancestor. We may be all netted together. That speaks to the expanding view that animals have rights, that animal suffering is important, and so forth. But the most important, what you might call the cogito, to sort of reflect Descartes, who said, "I think, therefore I am." Darwin said, "I think," and then drew a picture, a sketch of an evolutionary tree. And this is the one that you will see reproduced very commonly in the iconography of evolution. And in fact, some people, are so devoted to the cause of biology and evolution that they have that tattooed on themselves. And there's a guy called Carl Zimmer who's written a book called Science Inc. about scientists who have tattoos of science put on their own bodies. And here's someone who was so taken with that he had that put on their flank there. And Darwin's ideas were culminated in this book on the origin of species, which was published in 1859 after 13 months and 10 days hard labour, as he put it. And the key point is that you think of Darwin's theory of evolution as just like this one idea. He's just had the idea of evolution. But there was a whole conceptual backdrop, lots of different ideas fed into Darwin's theory of evolution and led him to publish The Origin of Species. And within that theory of evolution, there are all sorts of ideas all bubbling away beneath the surface, and he argues about all of these and provides evidence for all of these things in The Origin of Species in its individual chapters there. If you want a very short summary, this is a guy, Ernst Mayr, who is one of the great evolutionary thinkers of the 20th century. He actually lived over 100 years. He's sort of born kind of with living memory of Darwin and died only a few years ago in 2005. And he basically said that Darwin introduced the idea of branching evolution, common descent. Darwin said that the mechanism of evolution is largely natural selection. We'll see that's not entirely true as we go on. The idea that evolution is gradual, that suddenly, there's no rebooting of things. Basically, everything has to do it gradually. This is historical science, that we're kind of looking at what happened in the past and trying to recapitulate the past, restore the past in our understanding of things. That evolution feeds on variation and randomness, and for some people that's quite troubling. For a physicist, the idea that an electron is an electron, one electron stands for all electrons in the universe, all essentially the same, whereas Darwin's idea is that there is no essential entity, biological entity of any kind. So when the first human genome was published, it was that of a rather controversial figure, Craig Venter, and people said, shall we say Craig Venter is the type of humans, the central person? And everyone said, no, that's mad. And in fact, there's no human genome. There's, in a couple of days' time, isn't it? There'll be 8 billion. Is it 8 billion? It happened last week. There's 8 billion human genomes out there. And the other issue that happened here was that Darwin basically got rid of this idea that you can take a type and say that is the type, and that determines what happens. Anyway, that was just a brief aside. I hope you'll forgive me for speaking about Darwin for a few minutes. But let's bring it back to the subject of homology. So what Darwin did was basically redefine what homology meant. And what it meant after Darwin was that biological features in different taxa are homologous if they show similarity that is based on their descent from a common ancestor. So homology implies divergent evolution, or as the term that Darwin quite commonly used, descent with modification. And so having the idea of homology as showing similarity because of common descent gives us an explanatory framework for when we look at, say, the skeleton of different vertebrates, and we look to see that the the patterns, not just of their limbs, but if you look at the vertebral column of the skull, you can see these kind of deep similarities there that may not reflect current function, but they do reflect common ancestry. And just, I always like to provide the evidence. You'll find there's all sorts of quotes out there from people. You drill back and the person never said it. Here's what Darwin actually said about homology. And he said that in all this great amount of modification, there'd be no tendency to alter the framework of bones or relative connection. So what he's saying is that although natural selection will be making functional changes to things, the underlying architecture, if that's working still, there's no reason to change it. So there's this kind of interesting dynamic tension here between two aspects of evolution. One is that things change through natural selection, driven towards some function, to optimize some function. But underneath it, there is just this variation on what was already there, and there's this signal that stands over time of what things used to look like. And as he points out, that if you take this far enough, you might get a general pattern of an organism become so obscured as to be finally lost. So this kind of informational signal from the past may actually eventually get obliterated. And one of the things that I found interesting 10 or so years ago when I wrote the Rough Guide and I read a lot of Darwin is that how much of what he said back then is still poignant and true when we talk about modern concepts like sequence evolution. He had no idea about sequencing, he had no idea about DNA, he had no understanding at all of genetics, but nonetheless, his insights still are with us today. He also pointed out another interesting phenomenon, which is that when we look at patterns of similarity, we can look at the patterns of similarity between one species and another species, but even within the same individual within a species, we can sometimes see similar patterns. So just look at your hand and your foot. You know, you've got 5 digits, you've got the same kind of, you've got the leg, 1, 2, many, and then 5, 1, 2, many, 5. You can say the arm and the leg are in a sense homologous to each other. He came up with this idea and discussed it. Now you can argue that we now would call this parology, particularly in sequence terms, but the ideas are there originally, not just in Darwin. Owen noticed this as well, but it's all there. And this is because a particular component is duplicated within the individual. And so although it may have a common evolutionary ancestry to begin with, you get a duplication and then things go off in their different directions. And so the arm and the leg, the hand and the foot are subject to different evolutionary pressures. And the fact we've got an opposable thumb and we don't have an opposable big toe is evidence of that. Okay, well, let's fast forward. So if we move over the 100 years since the origin, the key development, of course, was development of genetics and Mendel experiments on peas, rediscovery of genetics, key distinctions that eluded Darwin, which was that there's a distinction between the soma, the body, and the germline, chromosomes, mutations, and this modern synthesis in the 1940s of evolution and genetics, where the two ideas of evolution and genetics came together in the idea that evolution was changes in the distribution of genes within a gene pool over time. Of course, another key development important to what we're talking about is the development of computing. And so Babbage was himself a contemporary of Darwin. I think they met at dinner once. And he invented this analytical engine, which is considered to be the first kind of computer. Ada Lovelace wrote the first program. And then we got these developments over time with Alan Turing giving us Turing machines and showing this, doing computation in one kind of set of systems is equivalent to others and so forth. And then we got computers. Now, interestingly, the first kind of hint that we could actually apply this kind of thinking to sequences came in 1965, where Zuckerkandl and Linus Pauling, who's interesting because he's one of the few people that got 2 Nobel Prizes, they came up with this idea of using chemicals as a basis for molecular phylogeny. And this is the opening paragraph of their paper. And you kind of think, well, how have times changed? The idea that you'd start a scientific paper by quoting Hegel and saying that there's no other system better than aufgehoben, constantly abolished, and it's just that it speaks to the idea of natural philosophy as a synonym for science. And it's worth stressing that you are doing a PhD, which is a doctorate of philosophy. So the idea that we touch on philosophy occasionally is not too alien, but yeah, it's a weird thing, isn't it? So what he was...
Don't put that in your manuscripts.
I wouldn't start your thesis by quoting Hegel. You might quote Darwin, but if you do, make sure you've read Darwin. This idea was that you could transplant these ideas of homology, as we see in morphological terms, into molecules. So there's this, then becomes this parallel streams of morphology and molecules as evidence for evolution, as evidence for the evolution of function, and even as evidence for what a function a thing might have. And it's an interesting sideline that Darwin produced all this evidence for evolution in the origin, but there is all this evidence in molecules. We could recapitulate the whole story of evolution just from looking at molecules without ever looking at a skeleton of an animal or the morphology of a plant. In 1968, an interesting insight came in, though. This guy, Moto Kimura, Japanese scientist, actually said what actually is happening when we look at sequences is that most of the changes we see don't actually make any difference to function. That it's just neutral mutations. It's just kind of noise, spelling mistakes in evolution, if you like, that don't make much difference or make any difference to function. And we'll say more about what that means, in terms of degeneracy of genetic code in a moment. And the interesting insight that came out of that is that those changes, those neutral changes, they show a clock-like behavior. So if you wait one period of time, you get one change. You wait twice as long, you get two changes, three changes. And so you can actually start to date when the various divergences of molecules happened over time. And this is not just the basis of molecular evolution, but it's also the basis of genomic epidemiology. And that will be a major theme of next week's stuff, is that you can actually understand how a pathogen has spread by looking at these changes. Changes that don't make any difference to how the pathogen functions, but just give you a molecular clock. In the late 70s, basically, we put our foot on the accelerator because Fred Sanger, another person who got 2 Nobel Prizes, actually invented DNA sequencing. He invented a form of DNA sequencing that took off. There were others who had invented other forms of DNA sequencing that didn't take off. But this rather humble Englishman who worked in Cambridge came up with the idea of DNA sequencing with chain terminating inhibitors. And he went on and did first a little bit of sequencing and sequenced a bacteriophage. And then within a short period, he went on to sequence what we might call the first bacterial genome, because mitochondria are, in fact, bacteria, if we look at their ancestry, and gave us that first mitochondrial genome sequence. And then, as we fast forward, we get into the 80s, and then molecular evolution of bacteria joins the great tree of life. And this guy here, Carl Woese, gave us a large sketch map of evolution in the round and said that actually the 3 main life forms, 3 big divergences. It wasn't just bacteria and eukaryotes or prokaryotes and eukaryotes, it was bacteria, archaea, and eukarya. And I've made this point before, and I'll make it again. It's kind of reminiscent of Darwin's original sketch, actually, of how evolution worked. And if you look in the notebook and you work out the invisible ink there, you can see that Darwin actually sketched this 150 years ago or more. This trichotomy here. And what this allowed people to do was to systematize evolution. So initially trees were drawn on the basis of morphology and And evolutionary thinking was really deeply embedded in that with a process called cladistics in the late 20th century. But since the 1970s, we've been drawing trees on the basis of molecular sequences. And there has been this clash between morphology and molecules. This is on the big picture, where, if you go and look at how plants are classified or animals are classified, there are all these people who say, oh, well, we do it by looking at their behavior or their morphology. And then a lot of people say, no, we do it by sequence, and the sequence tells us you've been getting it wrong the last 100 years. There's a lot of that going on. In bacteriology, we didn't have that because basically bacteria have so few features that trying to classify them in evolutionary terms without sequences was a fool's game anyway. But one of the interesting things, I'll just put up that paper again, is that there are many ways to create a text or a protein. And so if we see similarities in sequences, we can start to infer common ancestry in the same way as if you turned in an essay and you turned in an essay, and we looked at the precise form of words and we realized that they actually showed deep similarity, we'd say this is not coincidence. There are many ways to write an essay on sequence homology or whatever. They must show common ancestry. You must have taken your essay from her, or vice versa. I'm purely using this as I'm not suggesting that either of you would ever be engaged in such moral turpitude as to engage in plagiarism. But that's the way that plagiarism detection software works. It looks for statistically significant similarities between bits of text to show they have common ancestry. And of course, it may be that neither of you actually wrote the essay at all. You bought it from an essay bank. And handed it in. But basically, it's a very similar, and there are many of these kind of similarities between different ways of thinking. Now, I told you, you have to indulge me. For me, the moment in my career that was the defining moment was a particular day, the 15th of September, 1989. And I rather fancifully put up a picture here by Wright, Joseph Wright, showing the natural philosopher discovering phosphorus as an example of discovery, or quoting Keats here on first looking to Chapman's Homer, which I think is the poem for anyone who does science, really, because that thrill of discovering things. What I did was I looked a guy in the lab had been trying to clone an enzyme, the gene for an enzyme from Helicobacter pylori, which had only just been discovered a few years before. It was a gene for urease. And he had no idea whether he'd got it or not. He had a very crude antibody that had lit up a lambda plaque, and he'd subcloned it, and he said he had no idea. And he said he didn't get any functional urease activity from his clones, but he was just going on in hope. And I said, well, we could look at your sequences. He said, all right then. I said, are there any other known ureases out there where we have a sequence? He said, well, there is, but it's from a plant. It's from jack bean urease. I said, I just bought an Apple Mac, the very first Apple Mac you could buy, and we just got our first, I think there was about a few hundred protein sequences in the database, and I did a homology search, and I came back to him and said, yep, we've got clear 50% identity at the amino acid level between what you've cloned here and jack bean urease. And for me, that was the moment where I could see that using homology searches like this wasn't just someone pissing around on computers and wasting their time instead of doing real science. It actually had a decisive impact on the scientific program going forward. And we got the first paper out, and in fact, they got a patent out of that as well. So again, I'm drawing analogies here to try and help you understand this. I mean, we can look at this in terms of languages. If you take a look at languages and informational macromolecules, they show this similar kind of thing. So if you look at European languages, you can see that when we say 1, 2, 3 in English, we say eins, zwei, drei in German, and deux, trois, uno, dos, tres. You can see that there's some similarities there. There's a kind of there's an N there with a vowel in front of it. There's a T or a D sound made at the front of the mouth. And then there's the similar thing again, but with an R afterwards. This is not just by chance. This is because these are all Indo-European languages, and they all share a common ancestor. thousands of years ago. And in fact, one of the, for me, the interesting example here was the decipherment of Hittite here, which was a language that flourished in what is now Turkey in the 20th to the 13th century BCE. And this guy, a Czech guy, Bedřich Hrozný, looked at some cuneiform writing there where it said Nu Ninda, and I won't read it out, it's a tiny, Watama Ekutini, and Nu Bredna. Now, this Ninda was a bit like an emoji. It was an ideogram that was borrowed from the Sumerian language, and he said, oh, that means bread. You know, it's like a picture of a loaf of bread, if you like. So he could see it said Nu bread something wata ma ekutini. And this gives me a shiver down the spine. If you look at wata, it's clear. It means water. Same word today in English, Wasser in German. So over thousands of years, this similarity has been retained, and he was able to show, for some reason it's decided to move on. I don't think I told it to do that. But, so he was able to say, well, actually that means something or other bread, something or other water. He looked at the other words and he said, well, that looks a bit like the word for eat, and that looks a bit like the word for drink, or at least ekutini, he said, looks a bit like aqua in Latin, which means water. And he pieced it together. And in fact, even nu, I mean, in Hittite, it doesn't quite mean now, it's more like a kind of and word, but it is cognate with now. Cognate is what you say when you're talking about languages as homologous in sequences. So this is just an example of how you can recapitulate things. Now, let's get to the central topic of sequence alignment and homology. So what we do when we draw conclusions about homology is we rely on aligning sequences. And so what that means is that you're saying this particular residue here at position 1 is the equivalent to position 1 in the other sequence. The one at 2 is the position 2. Now, if you have identical sequences, it's obvious, isn't it? You just line them up, they all line up, it's fine. You don't have to do anything. And if you took an E. coli sequence and a Salmonella sequence, there will be some sequences where they are exactly identical. But the problem is that sequences diverge over time. And so what we need to do is devise methods for alignment, and if we're going to go on to search, we need to look for variability. And there are two kinds of variability. So one residue changes into another. So you can see here a G has turned into a C, or A as alanine's turned into a serine. Or actually you get insertions or deletions. So we call those indels for short, because often we don't know whether it's which way it's gone, whether the original ancestral sequence had that thing in and it was deleted, or whether it's been inserted in since there. So we call them indels to be kind of... and so what we have to do is we have to come up with a way of saying, well, we account for gaps going in and we account for substitutions. Now the trivial point, the reductio ad absurdam, is if you allow for gaps in sequences, you can align any sequence with any sequence. Because if you say you've got a bunch of T's and a bunch of G's, you just align all the T's with a load of gaps at the beginning of the G sequence, and the T's with a gap at the end of the G's with a gap at the end of the T sequence. You've aligned those sequences, but it tells you nothing. It's trivial and stupid. And so we have to kind of put some kind of penalty on gaps. Now, a key point is that when we look at sequence alignment and logic, amino acid sequences are more highly conserved than nucleotide. So this goes back to this idea of neutral evolution as the predominant form. Because the genetic code is what we call degenerate, so there are for most amino acids, there are multiple codons that encode that amino acid. And in many of them, it's the third residue that it can be variable, completely variable. In some, there are, in fact, multiple places in this table where they can sit. So you can look at serine up there, and you can see some serine down here. You can see arginine over there, and also arginine down here. So you can have exactly the same amino acid sequence. And let's just say you only mutate the third codon. Yeah, you can get 4 out of 4 amino acids identical, but 8 out of 12 nucleotides matching. So still quite a considerable divergence. But when you allow for the fact that serine and leucine, for example, can have completely different kinds of codons, you can get to a situation where, although the sequence at the amino acid level is entirely the same, at the nucleotide level you've only got 5 out of 12 matches. And so what this means is that a lot of the evolution we see is just this random noise of things that don't make any difference, which is informative in terms of trying to draw evolutionary trees, trying to do genomic epidemiology, but doesn't have any impact at all on inferences about function and what things are doing in terms of function. Whereas if we look at conserved amino acids, we can also infer some idea about function. I'll say more about that in a moment. Now, I'm going to talk for a couple of minutes about things I'm not comfortable with. I'm not a mathematician. Your first degree was in maths, wasn't it? No. What was your first degree in?
Software engineering.
Software engineering. So he's more comfortable with this than I am. But basically, how do you derive algorithms for doing this stuff? So one way of looking at, if you want to try and align sequences, you can draw what we call a dot plot. So you draw a graph like this, and you put a spot wherever there's an identity between the two sequences. So if we look at the first one there, A, and we try and align it with there, we can see that there's an A at position 2 and a position 4 there. At C, there's only one mix, one hit there. So there's at 2 at both positions there. If we're reading that one going up there from there, we can see that they align at 2 and so forth. So what you can see is that there's a signal along the diagonal there. And if you draw this over a whole length of sequence, if the sequences are very closely related or identical, you'll see a diagonal like that, a easily visible line, just the occasional discontinuity where there are gaps. Where there's more distant homology, you still get signal, but it's kind of broken up, and the dot plot will still show you, oh, these things are related, look, there are long stretches where they align, but there are also long stretches where they don't align and there's no signal. And that's typical of what we might call distant homology, where there's been a lot of variation over time. Now, I'm just going to quickly take you through this for those of you interested. As I say, so I'm not a mathematician or even a computer scientist. But if you wanted a simple algorithm, you could turn that dot plot into an algorithm by penalizing movement away from the diagonal, which is basically telling you that if you put a gap in to move it away from where position 1's lining with 1, 2 with 2, 3 with 3, and so on, there should be a penalty to stop you doing that. So you add a little penalty for doing that. And then the other key point about the way evolution works is that if you have a situation where there's two sequences and you need gaps to put in to align them, you look at the parsimonious principle of how many events have happened in the past to make this happen. And the most parsimonious explanation is the fewest number of events. The most common event would be that you would have, what's going on here? Why is it not? Oh, there we go. So that we have a situation where we could align these sequences like that, or if we just put in one gap, we get a much better alignment. And that is what happens in life, so that if a piece of the genome snips out, it might take out 10, 20, 100, 1,000 base pairs. It's a single event, and so it is common evolutionarily terms for that to happen. And it's far more common to, say, lose 1,000 base pairs in one event than it is to change one set of 1,000 base pairs into another set of 1,000 base pairs. So you have to take that into account. And so you have, what you do is you apply a low penalty for opening that gap, for extending that gap, but you have a high one to open the gap. So opening the gap, you say, no, we'll penalise you for that. But once you've got that gap, we'll allow that gap to grow a bit more. And as you go through this, you end up You also need to sort of have an algorithm that matches the letters that are better than mismatches, and you have a scoring matrix, and I'll say more about that in a minute, and then you put the two together and you end up with a scoring mechanism. Now that's me with a lot of wave of the hands trying to give you an idea of a glimpse of how these algorithms kind of work. You basically do all that and then you pick the route from the end back to the beginning that gives you the highest score, and that's called the Needleman-Wunsch algorithm. Another issue that we have to address is that if you try and align the whole of one sequence with the whole of another sequence and say, right, this sequence begins at 1 and ends at 100, this one begins at 1 and ends at 200, do you try and stretch out the one that's 1 to 100 to reach the whole of the 1 to 200, or do you say, no, the best thing to do is to try and align that 100 with the best 100 within the 200, if you like? So there's this distinction between global alignments and local alignments, and that's something else that's taken into account. Another thing that we have to take into account when we're looking at sequences is that we tend to use amino acid alignments primarily when we're looking particularly at function of proteins. And it turns out that not all amino acid changes are equivalent. So I'm assuming you've all done a little bit of biochemistry, so you're kind of vaguely familiar with the idea that there are different ways in which you can categorize amino acids according to their functional properties and their structural properties. So some of them are small and some of them are large, some of them are hydrophobic, some of them are polar, some of them are charged, some of them are aromatic, some of them are aliphatic, and so forth. And so if you wanted to change a leucine, for example, into an arginine, you kind of got quite a long walk in functional terms. You're walking away from the aliphatic group, moving through the hydrophobic groups, skipping across into polar residues, and then finally ending up in positively charged ones. So you're making a lot of changes to function. And so you could imagine that would actually be something that you'd want to say, well, that's unlikely to happen. And in fact, you can look at it from that point of view of like how many functional changes, but you can also look at it, evolution's done the experiment. If we align proteins that are homologous and we look at the patterns of substitution, we can say, oh, these kind of substitutions happen all the time. They are, that's a very common thing. Whereas these kind of substitutions are actually very, very rare. And if the alignment's telling us that's the kind of substitution we're getting, there's something going wrong here, or it's not, or it's very, it's hard to. So an alanine turning into an alanine, you've been retained as an alanine, you can give that a score of 4. Cysteine being retained as cysteine, a 9. So you get these scores on how much things change. And interestingly, in this is the so-called Blossom 62 matrix. This is one of the commonly used matrices in homology searches and in alignment programs as well. You can see that if we take some of those changes there, so an E into an E gives us a score of 5, a D into a D gives us a score of 6, but an E into a D actually still is very common because these are functionally equivalent amino acids, aspartic and asparagine. The glutamate and glutamic acid. These changes happen all the time in evolutionary terms, and so and similarly, if you've got alanine, which is a small amino acid, and glycine, which is even smaller, they turn into each other all the time. So in some cases, you've got positive scores to say, well, yeah, that's perfectly allowed. Other cases, it will be penalised. Now, in terms of talking about homology, we have to just revisit the idea that homology is not the same as similarity. And a lot of people will talk about similarity homology as if they are the same, but in fact they're not. So homology you've got similar features because of common descent. We usually cannot know what the ancestor looks like. So this is a kind of hypothesis, if you like, or it's an inference. Based on similarity. Likeness, the similarity is just this is the amount of similarity we see. And you can use statistics to say, oh, this is highly similar, highly significant similarity. And we actually do use those kind of statistics to determine homology, but they're not quite the same thing. And there are some people, again, if we're thinking about you in the Viva and you've got a grumpy old professor on the other side of the table as your examiner, some people say that you must say that homology is either there or it's not there. And you can't say, like, these sequences are 50% homologous. You have to say that they show 50% sequence identity. Or if you allow for a substitution matrix that defines what you mean by similar, you could say, oh, well, they show 50% similarity. But if you say 50% homology, you get your knuckles wrapped. It's one of those things. It's a litmus test that some people will say, oh, if you say 50% homology, right, that's it. You've crossed a boundary, and the rest of this Viva is going to be hard, not easy for you. I'm going to make you suffer. This sounds childish, but these kind of events do happen. There's a turning point where if you flip the examiner to being your enemy rather than your friend, this happens. Some people make this analogy with pregnancy, which I think is rather fanciful, because it's perfectly possible to say someone's heavily pregnant. It's not just an all or none thing. You're either pregnant or you're not. You could be heavily pregnant, but it's a kind of interesting point. Anyway, I better move on because we're going to run out of time. So when we do homology searches, what we're trying to do is we've got a sequence, a query sequence, and we want to search a large database of sequences. In fact, often what we want to do is we want to search all known sequences available to us, all the ones that are, I still think this is astonishing that you can do a homology search across the accumulated knowledge of humanity on proteins, and you can search your little one sequence against all of that stuff. And what we commonly use, and we're going to talk about in a moment, is using the BLAST suite of programs, and I'll say more about those in a moment. We end up with a significance score. So BLAST is the kind of industry standard for this. Stands for Basic Local Alignment Search Tool. First published by Altschul et al. And I haven't checked lately, but this is one of the citation classics. Tens of thousands, maybe hundreds of thousands of citations to this paper. It stands up there on a pedestal among the most highly cited papers in science. Not going to go into lots of details about algorithms, but what happens here is they kind of make a word list of exact sequences, matches, and high scoring substitutions, and they search through the database sequences for things that match. And then if they find two hits that are on the same diagonal alignment, they extend that until they reach a certain threshold and it drops off. And they create what they call highly scoring segment pairs. And this approach is what we might call heuristic. It's not guaranteed to give you the perfect best answer, but it works pretty damn well. And it works much more quickly than any exhaustive search would do. So there's a huge increase in speed. And so I still, we get the anaesthetic of familiarity of the self and Bill and others when we do homology search. But if you step back, wow, you've searched the whole cumulative knowledge of humans on proteins in a few minutes, and you got a result. Wow, that's just amazing, I still think it is. So when we look at these results, and we'll look at some in a moment, what's the evidence for homology? Well, we basically want to look for similarity that extends over a long stretch of sequence and is statistically significant by criteria known to be reliable. So one of the things you can do if you're a bit bored later on, you can put in your name into a homology search and search for it. And if your name is spelled out in the single letter amino acid code, you will find matches probably. And you all did the the ones on the MMB course did the aptitude test where you saw the name Darwin spelled out in single amino acid code. You can do homology search and you will find that string Darwin in proteins. But that's because there's so many proteins out there. It doesn't have any meaning whatsoever to find Darwin in a protein. It's just what you're looking for is longer stretches where you can say, wow, that means something. And even if when you get to kind of fairly low similarities or sequence identity scores, if you do multiple alignment where you line up multiple things that appear to be homologous, and you can see that there are certain things that really are conserved across the whole thing, that's evidence of homology. If everything's just random, one thing's showing a bit of similarity to the next thing and then to the next thing, but there's nothing that's conserved among all of them, then it's like this could be noise. And what we tend to find is that those things that are most conserved are those things that are functionally important or structurally important. So when people have done experiments, they can say, we know that's the active site of that enzyme. Or when they've solved the structure, they say, well, that's a particular bit where that feature and that feature get very close together. There's no space for anything else apart from a glycine there. So you'll see these similarities, these homologies that actually, it's not just this boring old, just reflects evolutionary ancestry, so what? It's actually telling you something important about the function and structure of that particular protein. And when we're looking for this, we're not just interested in the score of the alignment, but how likely are we to get that alignment by chance alone? And this is where the so-called Carlin-Altschul statistics are used, where they basically say the chances of you finding, like say Darwin, if you took Darwin and you searched a database of 100 sequences, back in 1980s when I was searching databases of a few hundred sequences, and you found Darwin in there, you think, my goodness, there's a signature there that natural selection actually recognises the importance of Darwin or some such weirdness. It's not by chance. But now, where there are, in NR there will be millions, tens of millions of sequences in there, the chances of finding Darwin by chance alone in that data set doesn't mean much. And what we tend to use is something called the expect value, or the E value, which is the probability of obtaining this score by chance. And it's the best measure of how biologically significant an alignment is. And what these programs do, like BLAST, is that they will go through and calculate the E values for all those matches, and then they rank order them, and they present you the most significant E values. And it's one of those things that the lower the E value, the more significant it is. A low E value is evidence of homology. So BLAST as a default uses an E value of 10. That's a very, very flabby E value. That will give you all sorts of stuff that's probably not significant. The clearest evidence that you've got homology is when the E value contains an E. So it's framed as 2E to minus 19, which means 2 times 10 to the minus 19. And so if you've got that E in there, that generally means, this is homologous. I'm going to just pause for a second here. Has anyone got any questions or comments? Because I'm.
When you say value default 10, is that the highest? I'm a bit lost on that bit. It's like they show you everything that's
Yeah, so
10 and below.
10 and below in a sense. So if you did a search of the database with a sequence, it would show you everything from 0, you can get 0, where the score is actually 0, down to 10. And it depends, all this critically depends on the size of the database you're looking at and how similar whether your sequence is one that is already related to lots of sequences or completely new. But so we'll be looking at what BLAST shows you in a moment, but you can tell BLAST how many hits to give you. So you can say, I just want to know about the top 100, I want to know about the top 1,000, I want to know about 50,000. I think the most it will give you now is 50,000. And so if you get a score of 10, that is pretty borderline. It's not impossible. It's going back to Darwin's point originally, which is that you've got these two things similar. Over time, that similarity is eroded by descent with modification until such time that you can, although it's there, although they do have a common ancestor, you can't find any evidence for it because it's been eroded. So 10 is, you know,
Is 10 the number of hits that you can expect by chance?
by chance in a database of that size.
In that database
Yeah,
of that size.
So if you get
And-
50 hits
Yeah.
with a score of 10, you could say, well, that's beyond what you'd expect by chance. But this is at what we would call the twilight zone of homology, and it's it's an interesting place to explore. And I've some of my most highly cited papers I have been scrabbling around in the twilight zone, but it's slightly a dangerous place as well because you can infer similarities that are not there. I remember when I was a schoolboy in French, we learnt the word quitter in French, and I said, "Oh," to the teacher, I said, "Sir, is that related to quit in English?" He said, "Yeah, it is. Common ancestry, yeah." The next day I said, "Oh, I've just seen this word hiver, means winter. Is that related to shiver?" It's the same word, but with an S on. No, completely spurious. And that's the sort of thing, you know, it's as basic as that, really. We're looking for, any of you who've learnt a foreign language, you come across these so-called false friends as well, where the meaning changes. We do see that in evolution, that although there's similarity, it doesn't mean that they're doing the same thing. Anyway, let's get on.
You mentioned the e-value will be the database size mark saying
And the length of the
and the
alignment.
length of the sequence.
Yeah, yeah.
I don't think it's not on the slide,
Mm.
but yeah.
So in terms of doing homology searches, rules of thumb for homology searches, as I mentioned, the sequence homology is best conserved at the amino acid level. So if you're interested in protein coding genes and in proteins, you search at the amino acid level. Obviously there will be some cases where you can't do that. If you're looking at tRNA, for example, or
Just any intergenic region.
any intergenic reason that's not translated, then that doesn't work. Now the databases do actually allow you this quite cool trick. So the BLAST suite allows you to actually translate the query sequence. So you can take a nucleotide sequence and it will translate it on the fly into all 6 reading frames and throw that against a protein database. Now the problem with that is it's computationally intensive to translate everything on the fly in 6 reading frames, but it does work. You can do DNA versus DNA. For protein coding genes, if you were looking at E. coli and Salmonella, it's reasonable to do that. If you were looking at E. coli versus human, you were looking at some ribosomal gene, and you tried to do that at the nucleotide level, you would not get a signal. You would just about get a signal, maybe for some of the ribosomal proteins at the protein level, but you wouldn't. And then there is like I really can't be bothered. I'm tired. I don't want to do any work at all. Just let the computer do all the work. You can get the computer to translate your nucleotide sequence and to translate all the nucleotide sequences in the database on the fly and compare them at the amino acid level. That is a very computationally intensive process. And if you try and do that with we're going to use the NCBI suite, most of the time it will say, go away. You've exceeded your quota of computational allowance, and it just doesn't give you a response. Now, I coming to an end soon. Where are we here? We did start a bit late. One of the problems we've got with homology searches is simple sequences. So many proteins will actually contain regions of very low compositional complexity. So those kind of substitution matrix where we say, oh, the chances of an alanine turning into a glycine or a proline turning into a phenylalanine is this. Those and the scores you get from them are based on the idea that you're drawing on the full alphabet of amino acid sequences. But if your sequence is just all glycines, you've got 50 glycines in a row, that will look very similar to any other sequence that has 50 glycines in a row. But in evolutionary terms, the ability to just come up with 50 glycines in a row by chance is actually quite substantial. It's possible for that to have arisen more than one time in evolution without it being evidence of is what we call convergent evolution. There's no evidence that they're actually similar as a result of common evolutionary ancestry. So we get this problem where some of these sequences show similarity not indicative of homology. And what happens is that the programs actually then use these so-called composition-based statistics. So they take into account, so hang on, these two sequences only contain glycine, or they contain 50% glycine, which is completely deviant, and they address it here.
Usually they mask them out.
Well, there's two approaches. One is you can mask them out, so you just exclude any regions that have huge compositional bias and say we don't bother to even look at that. Or when you do look at it, you apply special statistics. And one of the things you'll find, and you'll be going on to look at predicting protein coding genes in a genome, is that you do often get little short supposed protein coding genes that have this great compositional bias. And probably they're all artifacts. They're not real proteins. They're not ever translated. They're not real. And this is one of those interesting things. Is that if you, some of them are, and this is the interesting. If you say to me, "What's the smallest protein coding gene?" And, you know, could you have a gene that only had a methionine, and it made a protein of one? Well, no. It had a methionine and one other one. Could you have a 2 amino acid protein that was actually made from a gene? And where do you draw a line? You know, so these are interesting points. And here's just to make it concrete. Here's some examples. So here's a couple of proteins that are proper, you know, they're proper proteins. They have within them what we call globular components that actually fold up into something. But within them, they have these segments that are actually greatly compositionally biased. So these are often called Q linkers. So if you did a blast search with either of those and you left it without any corrections, they would find out everything else, including this one down here, that had all those Qs in. And it wouldn't mean anything, made no inferences at all about common evolutionary ancestry or anything like that. And so, as Nabil points out, they would be masked out as you did the search, and if you left them in, the composition would be taken into account as well. And this is an example of a protein that I mentioned. Like, this is a hypothetical protein that's been predicted from a bacterial genome. I don't believe it. I mean, as Nabil is playing devil's advocate, it's not impossible in biological terms that a protein like that could exist. But I'd be strongly skeptical to say, well, that's probably either it's an out of frame translation, and if you go to another frame, you might get a more sensible protein, or it's not even a protein coding region; it's an intergenic region that's been misidentified. This is going on longer than I expected, but we're nearly there now. So I mentioned orthology versus porology. Another interesting question that arises is that when we look at these similarities from sequences, we don't necessarily see them between one organism, just see them between one organism and another. So a gene in E. coli or a protein in E. coli shows homology to one in Salmonella. But within E. coli, you can have multiple proteins that are homologous to each other. And that is what we call parology, where there's been a gene duplication in the past, and those two genes have been retained in the genome, but they've diverged in their sequences. And sometimes one of them will retain a characteristic function, and the other one will diverge. Sometimes both of them diverge from the ancestral sequence. And here's an example from human, vertebrate evolution that we know. We had early globin gene, then it duplicated, and you end up with an alpha chain and a beta chain, and we look in our genomes and we can see we've got these different paralogs and homologs. And we use the term ortholog for those that where they show the common evolutionary ancestry within that genome. So it's a slight twist on the tail here. Has anyone got any issues with that? You kind of understand where we're coming from? I mean, the same. We see similar patterns in languages. So in English, we had the word regal and we have the word royal, or legal and lawful. So and legitimate. You know, they all have common ancestry, but they're all in the same language. Now, what we want to do when we've got a genome is we want to annotate that genome. I did have a friend in the medical microbiology department when I said, we look at genomes. And she sort of thought, what, so you sit there and you just go A, C, T, G, G, C, G, you just look at the... None of us are like the human computers, we can just look at a sequence. We have to actually analyse that sequence and annotate it so it makes sense. And one of the things we do is we call out the protein coding genes, and we'll be saying more about that later. But once we've done that, we then try and annotate those protein coding genes. And we tend to do that by using homology as a guide. But one of the problems with homology in this context is that proteins they're not just lumps, that indeterminate lumps that can't be broken down. They consist of domains, actually, that are strung together. Functional components, structural components that are strung together. And if we're drawing inferences of homology, there is this interesting point, the so-called transitivity of homology. So if we can show that A is homologous to B and B is homologous to C, we can infer that A and C are both homologous as well. They all share a common ancestor. So that's interesting. And we can abstract out the patterns of similarity that we see between those three and use that to find even more distant homologs. But one of and if you like, when you try and solve what a protein is doing from looking at its gene sequence and the protein sequence, it's a bit like solving a crossword puzzle, because you're basically trying to break it down into pieces, look how the various clues interact with each other, look at how homology informs your understanding. But one of the problems that happens is that if you just say, right, what I'm going to do when I annotate the gene in organism, I'm going to take the gene in organism A and I'm going to use homology to that to make annotation of the gene in organism B, is that if you choose the wrong part of the protein and make inferences on homology based on the wrong part of the protein, you end up with this so-called annotation catastrophe. So if you look here, one of those proteins of that set of 4 proteins has got a toxin domain in it. Only one. But because they're all got homology somewhere in the middle of the protein to each other, they're all, with a wave of the hand, you say, they're all homologous to each other. And if you carry that, say, all homologous, then you would take, if that was labelled toxin, you might take the toxin label and just smear it over all of them. And so this is the kind of problem that you get with homology inferences in terms of annotation. And we have for a long time now been in a situation where the number of proteins that have been annotated by homology vastly exceeds the number of proteins that have experimentally or structurally verified features. So even in an organism like E. coli that we've studied for over 100 years, I would think that the majority of annotations are still by homology alone. And so what that means is that if you really care about what that homology is telling you, you have to trace that evidence trail all the way back to experimental or structural evidence. We can take an example from Islam. So in Islamic tradition, Muhammad said a load of things when he was wandering around, and people said, Muhammad said this about that. He didn't write it down anywhere, but people said, well, what do you think Muhammad said? I remember on a Friday afternoon in the marketplace, he said, you mustn't do that, or you must do that. And this is called, what they have in Islam is an evidence trail, which says, well, if the evidence how do we know that Muhammad said that? Well, we know that because this guy who lived at the same time as Muhammad, was a contemporary of Muhammad, said that he said that. That would be very strong evidence. And this is called hadith in Islam. I'm sorry to talk. Is this okay to talk about this? Yeah.
It's fine so far.
Is it? But if you end up with a person that you say, well, this hadith here, what's the evidence for that? Well, this person here who says that this person here who happened to know Muhammad said that. Well, okay, the trail's getting a bit long now. And in fact, it turns out that the first person, second person in this trial and the third person didn't even overlap in that they lived in different cities and different times. So how can they ever have passed on that information? This is unreliable. And it's the same kind of argument here that I've made the fanciful point of genomic hadith in the past, that we need to actually follow these evidence trails if you really care. Now you might say, well, and this is the issue now, that you sequence a genome, 1,000 genes, 2,000 genes, and I haven't got time to go and care about every one of them. And the overwhelming majority of the time now, annotation just gets sprayed over by homology, because from homology searches. But there may come a time when, if you're working on pathogen biology, that you will actually study one of those genes in particular. Oh my goodness, this gene here seems to be crucial to the thing I'm looking at. Nobody knows what it does, and you start looking at homology, and then you'd care, and then you have to go back and look at these evidence trails. Right, that's me in the talk and chalk mood. I was now going to take you through, I've prepared slides for it, but we can do it in real life. And you can actually follow me if you want as we do it. We're going to go to the NCBI website. So has anyone been to NCBI before?
I hope so.
Has everyone been? Anyone not been to NCBI before? Nobody wants to admit to it. You've been there? Yeah. I mean, you've been there because it's the home of PubMed, probably, primarily. It is the most awesome site on the planet. Whatever we think of America, its gift to humanity is the NCBI website and the suite of programs available to us there, which are made available to all of us for free of charge. And because they're produced by the federal government, no copyright is attached to them.
It's all public domain.
This is one of the key examples of we the people. Okay, so this is, I'm just getting a bit politically waxing lyrical there. And we can go to NCBI and we can search for my name. So why don't we do that in real life and come out of the PowerPoint.